Drug developers, investors, researchers, and corporate executives wrestle weekly to understand what is happening in commercial development of NASH medications. Join hepatology researcher and key opinion leader Stephen Harrison, patient advocate Donna Cryer, liver wellness advocate Louise Campbell, and forecasting and pricing guru Roger Green as they discuss the issues affecting the evolving NASH market from their own unique perspectives on the Surfing the NASH Tsunami podcast.
Hi, this is Roger Green, executive producer and host of the Surfing the NASH Tsunami podcast, and welcome to our year-end review. Based on listener requests, we are releasing each of our 8 year-end interviews as what we call an extrasode, a 20 to 25-minute piece covering a single topic. This extrasode is with Vlad Ratziu, who provides a fast, detailed, and extremely erudite overview of major events in drugs and diagnostic development in fatty liver disease in 2020. Questioners include Louise Campbell, Stephen Harrison, and me. And we begin. This conversation is with Vlad Ratziu, who's joined us for our EASL and AASLD coverage and once or twice more throughout the year. And Vlad, welcome. And which event of 2020 would you like to focus on today?
So thanks for the invitation, Roger. It's always a pleasure to be here and discuss with you all. So obviously 2020 was a very special year with this pandemic that hit all of us and had a profound impact on both our professional activities and on the way we interacted with each other. And there's a lot to say about this, but obviously what we need to focus is on, in terms of NASH landscape, what happened during this year. So despite the bad things that happened, the limitations in terms of conducting clinical trials, this was a very good year actually for NASH clinical trials. I think that one of the major things that 2020 brought us is some significant advances in terms of therapeutics for NASH. We had the results of several trials this year. We had the unfolding story of other molecules that are in development for NASH. And all this makes for a very, very vibrant environment in terms of therapeutic research in NASH. So I'm very pleased about that. We had both results from phase 3 trials and results from early phase, smaller proof of principle trials with innovative molecules, both reported in the same year. So the field is clearly in ebullition here. It is boiling hot in terms of NASH therapeutics. So what we've seen this year is more data about innovative molecules, and some of them, like semaglutide, have been awaited for a long time. That was a long 18-month trial, reported positive results this year. We also have some disappointing results from elafibranor, a PPAR alpha and delta agonist, which reported a phase 3 trial that failed at the interim analysis. But at the same time, we had very promising results of another PPAR agonist, which is a pan-PPAR, lanifibranor, which showed in a much smaller trial of shorter duration, surprisingly good histological results. So a lot of progress along the road with some older molecules that confirmed or not their efficacy, and some newcomers, which are very interesting to look at because they come from classes of drugs that are already well established, and yet for which it is important to understand how much more they bring to the table than the first-in-class compounds. And for instance, I'm referring to FXR agonists, and we had results in regard to that, tropifexor, which is one FXR agonist. We also had very, very promising results with other classes of drugs, such as the FGF21 drugs, and now there are several in development, and some of them look very interesting, and here again, a lot to study, because there are significant differences between these compounds, not only in terms of pharmacological structure, but also in terms of what they have shown to be able to achieve in terms of clinical results. And then close to this, we have aldafermin, which is an FGF19 engineered compound, probably closer to the FXR agonist than to the FGF21 agonist, but which also continues to provide year after year very interesting phase 2A and phase 2B results from smaller trials, trying to fine-tune the dose, and each time with very, very promising results that are each time confirmed by all these subsequent trials, confirmed biochemical efficacy, and then some additional hints of very strong histological efficacy as well. So a lot of new classes of drugs, different stages in development, very promising results. We now take for granted the fact that these trials can be conducted and completed. But I remember 10 years ago, very few people would think that we could do NASH trials, finding these patients, doing the biopsies, following them long enough, having them to accept an end-of-treatment biopsy in order to measure the histological effect. At that time, very few people thought that that would be possible on a larger scale, more than once for one molecule. Well, it turns out there are a lot of these trials ongoing. And clearly the field has adapted to this medical need and to the specifics of the trials in this population. And we start now, hopefully, to reap the benefit. Now, there have been also some interesting reports in terms of how do we measure success in these trials, regardless of what drug is being used. And this has been published this year, very important papers on the limits of the pathological readings and how we should deal with that and what are the limitations that we should know about, but also confirmatory data, which is very important, about non-invasive markers and how these could measure therapeutic efficacy. Some of these markers, particularly the ones that are based on imaging, are now becoming mainstream in terms of trial conduct. I'm referring, of course, to all the MRI-based methods to measure liver fat content, or even now slowly starting to emerge the ones measuring fibrosis through elastometry methods. So all this makes for more efficient trials for endpoints that are better standardized, more quantifiable, and provide early results. All this should help drug development. So I found 2020 particularly rich with this type of data that hopefully will help us move towards the day when we'll have a first drug approved. There were some disappointments. For instance, the fact that the NDA by OCA, the obeticholic acid, was apparently rejected, at least in the first round, by the FDA. That was disappointing because it is unclear exactly why that happened, and in particular, we're all conducting these trials, and we continue doing so with an understanding that if you meet some of these histological endpoints and if you demonstrate the benefit, the way it was framed actually by the agencies themselves. And if you demonstrate that benefit with a sufficient margin of confidence in terms of statistical significance, that should give you the solidity of the data to be used to authorize a drug to be put on the market, or at least on the benefit side, it should give you sufficient data for that. And it turns out the situation is a bit more confusing now because clearly obeticholic acid had reached the efficacy endpoint with sufficiently large level of evidence, and yet the FDA for the moment rejected that initial NDA. So unclear. We'll need to better understand what it is about, and we'll need to continue working on optimizing the endpoints in the clinical trials so that it satisfies both the regulators, it satisfies the operational requirements of these trials, which can't run for 10 years with 500,000 patients in each arm, and which also satisfies the clinical relevance of what they measure. And at the beginning of the year, we thought all this was sort of agreed upon and already established, and yet we now discover that maybe there's more work to do on that. So I think those are the main things I would take away from 2020, and I really hope that we can make even bigger efforts, progress in 2021, particularly if we can go back to an environment when people can interact more easily, exchange ideas more freely together in physical meetings.
Vlad, thanks for a tremendously rich and broad description that covered a lot of territory and a lot of events, I think, efficiently and very, very quickly.
I would say this is terrific, your summary of 2020. And it reminds me of the parallels with NASH natural history, where we think 2 steps forward and 1 step back. And with drug development, I think we've seen the same thing. You know, we've seen some very positive movement forward, but we've had a couple setbacks along the way. And it just struck me that there was some parallels there. As we look forward to 2021, really the question is, how do you see it playing out? What do you see happening? Assuming COVID is what it is and we get vaccinated, and by the end of the year, maybe the second half of the year, we're meeting in person again, we're back to status quo. How do you see the field moving forward? It doesn't just have to be in drugs, but just, non-invasive testing, regulatory ideas. Where do you think we'll be? AI, artificial intelligence play, just some, putting on your futuristic hat. How do you see 2021 playing out?
Well, I think there will be some work to make truly important advances in biomarkers and someday replacing biopsy for the purpose of both clinical practice and clinical trials. So I see 2021 as a continuation of the previous years with a richer set of data demonstrating how we could use these biomarkers. We understand very well their limitations in the sense that they're being compared to liver biopsy, which itself has limitations. So therefore, you can never reach a perfect value of a biomarker because it's compared to an imperfect standard. That we understand. But there is accumulating evidence in favor still leaping over the biopsy and demonstrating the value of these biomarkers as an indicator of the status of the liver, of the prognosis of the patient. I expect more data in 2021. Particularly important, it will be the demonstration, now that we have these large data sets from phase 3 trials, that patients that are labeled responders based on histology are also the same patients where you can see changes in biomarkers that are meaningful, and therefore by using that, we could define response based on those changes in biomarkers, knowing that they have been sort of correlated with histological changes. So I think that will be an important thing that will happen in 2021, because now we have the data. We have data from OCA, we have Regenerate, we have data also from Resolve It and other trials, semaglutide. So I think 2021 will bring us one step further to using these biomarkers as a parameter that determines response in clinical trials. 2021 will also be a very intense brainstorming year in terms of how we use pathology in clinical trials. So that is traditional pathology and how we optimize this. And you know all the discussions around this with a number of pathologists, the strategy to read these slides. But also in terms of automated aids to pathology, to traditional optical pathology, and this is where the artificial intelligence type of automated reading will come in. And I'm sure 2021 will bring much more information on that. So a lot of this information comes from people doing specific studies, but most of it actually comes from pharma and sponsors that integrate these methods to their clinical trials. This is what happened with MRI-PDFF, and you see that now it's a well-accepted biomarker. I think it might happen the same with AI, but probably the issue with AI would be to sort of standardize and decide what type of approach you were using. MRI-PDFF is just one unique single tool. AI can take different shapes and forms depending on who is developing it. In 2021, I think that the most urgent things we need to work on. And then, of course, all the studies on natural history, understanding the heterogeneity of the disease, the groups at risk. There's still a lot to do. It's still not entirely clear what we do with people with moderate fibrosis, such as the stage 2, F2 people. Should they be treated? Should they not be treated? Are they truly severe? Which one of those will progress? Those type of studies need to be fine-tuned, refined, and we need to have a much better understanding of this intermediate category of patients. So those will be some of the things, but I certainly cannot think now of all the important things that will occur. And maybe, Steve, you'll make a big discovery in 2021, which everybody will be talking about at the end of the year.
You've mentioned so many things, and as I was sitting here listening to you, I was blown away really by how much work is currently being done in NASH. And it's so much more than just a drug to treat NASH, isn't it? It's identifying the right patients that need to be treated. It's developing the right drug for the right patient at the right time in the spectrum of their liver disease. And it's understanding how we move beyond liver biopsy to something that's simpler and easier to use. It's looking at non-invasive tools to assess efficacy of drugs and even begin to predict long-term outcomes. And so I just want a quick follow-up question. When we talk to Mazen, Mazen Noureddin has done quite a bit of work on MRE, looking at predicting an outcome benefit based on not an outcome benefit, but based using to predict an outcome, and really following on work done by Alina Allen at Mayo. We had a discussion on his segment where we mentioned that while MRI-PDFF might be used as a marker of therapeutic efficacy in drugs that move liver fat, for instance, some of the FGF21s, resmetirom, thyroid hormone receptor betas, and maybe the FGF19, could MR elastography be used in a similar fashion as MRI-PDFF for people with cirrhosis? In other words, as we study cirrhosis, and as we study drugs that impact cirrhosis, could we use MR elastography as a marker of efficacy that could then subsequently be used to predict outcomes?
I think based on the data accumulated so far, it is premature. But of course, all this can change with more studies and more data. The problem we have with MRE is that most longer-term phase 2b and 3 trials, most of them did not use MRE in their study design. So we won't have those data. And unfortunately, those data are very important because those are the ones that would associate MRE with histological changes, improvement or deterioration. So the only studies that have used MRE in trials, are the early phase 2A trials. And those are too short, first of all, to allow a biopsy to be done. And then they're too short to really meaningfully assess fibrosis changes. So I'm afraid that for the moment, we're a little bit blocked in a situation where MRE is not being used and studied at its full potential, at least in clinical trials. And most of these phase 3 trials are already underway. They do not have MRE planned because of the sheer number of patients that are included. It's probably something that's not feasible. So I suspect that it will be a little slower for MRE to reach the same point as MRI-PDFF did. Nonetheless, the idea of having elastometry as a marker, it's happening. It's materializing because you saw the data with FibroScan. It's poor man's elastometry, poor man's MRE, but it still provides data that can somehow be associated with histological changes. So that we have data. For MRE, I might be wrong, but I see this happening a little slower for reasons that I mentioned. And then individual studies done by people like you and me do not have the power and the prospective design to really show that MRE tracks with histological changes. We can have occasional anecdotal small series of patients, but we won't have the power that you have with a well-designed trial where people have this before and after. I don't know. What do you think? Do you think that it's coming of age for using trials in clinical practice?
Well, I think, as you alluded to, it's a work in evolution, and we need to generate data. And only after we generate data can we analyze the data and really apply where we think it will be best utilized. What struck me from the work that Alina Allen has done is that if you take a cirrhotic, for instance, and you have an MRE of 5 kPa, over 3 years of follow-up, that patient has a 20% risk of decompensating. Alternatively, if the MRE at baseline is 8 kPa, over the same 3-year period, there's a 40% risk of decompensating. So my thought would be, if we set a baseline MRE value of, say, 6, for instance, and we reduce it to 5, did we reduce that rate of theoretical progression of that liver disease? And if we do, we can look, for instance, at the work I presented at AASLD on an interim analysis of the first 123 people participating in the resmetirom trial that were dosed at 100 milligrams. And we saw a 50% reduction in MRI-PDFF in the 16 weeks. And we dropped MR elastography by 0.34, which was highly significant. But I don't know what that number really means. Like, I can look at the PDFF number and I can say reducing by 50%. Theoretically, that has some impact on NASH resolution and maybe even fibrosis improvement. But I don't know what an improvement of 0.34 in MR elastography means. And I think that's where we need to get more granular data in 2021.
I think that, just to say very briefly, I think applying this to cirrhosis would be very interesting. But remember, we have so much data on HVPG and prognosis in cirrhotic patients, and still HVPG did not really make it as an undisputed outcome in cirrhotic trials, despite all the massive data we have. So we need to keep this in mind. But of course, it would be great if MRE would be a good indicator of fibrotic changes in all clinical events.
You know, you and I could talk forever about these sorts of things, but maybe we should let Roger and Louise ask a question or chime in as well.
I only actually had one comment because it was a great summary. All my thoughts were, in the trials that were conducted in 2020, all of the participants were already recruited and known for most of those studies. So the ones that have been delayed, the ones that have been postponed, we have waiting lists in liver units and hepatology throughout the UK, and some of them have waiting lists of over 12 months now for patients. So the actual ability to locate patients in 2020 with the length of waiting lists, certainly in the UK and maybe around the world, does that pose a problem to identifying the right patients for the future trials?
How so, Louise? How exactly would that evolve to be a problem?
If you keep to your normal clinic schedules, whereby you're only seeing 30 or 40 patients in a hospital a week through a specialist hepatology department, and that doesn't change, and yet your waiting list is a year long, those 30 or 40 patients coming through may not be of quality for NASH studies. What you want is the pool of a year's worth of patients waiting, screened sufficiently quickly to be able to identify patients to go into the future studies. I don't think the volume of patients is going to come through in the way that it was before, because there was no real waiting list.
Yeah, but the question is, does this waiting list dilute actually itself in the large pool of patients who are not even recognized and diagnosed? That's a problem. But it is true that COVID impacted heavily in terms of patient management, particularly those for reasons other than COVID. And a lot of people missed their appointments. A lot of people missed exams that were supposed to detect cancer or monitor for cancer appearance. So it is quite dramatic in terms of negative impact in non-COVID diseases. I think that, at least from my point of view where I practice, I think it's manageable. The biggest problem we face is not so much having too many people waiting, because we adapt ourselves. We work longer, or we have more people that try to help us for that. But the real problem is not enough patients being referred to us, despite them having the risk factors for NASH. So the eternal problem of lack of recognition of the disease. I personally don't see here in France. So there were some delays, but on and off we've been working like maybe 80 to 90% of our capacity in the past 6 months. So it is a problem, but I think we will be able to manage it. And most of these patients are not like they need to be treated or managed within 1 month, right? You still have enough time to do the work properly and in a good way.
So Vlad, let me ask a different kind of question. There's a term called a blinding flash of the obvious. What that refers to is something that nobody sees, and then once it becomes clear, you can't believe you didn't always know that. Okay? Can you think of anything in 2020 that would qualify? A moment that when it became clear, it transformed all your thinking, both forward and backward? It could be any of the drug trials. I sometimes think about PDFF when I think about this, but where would you go?
If you frame it that way, it's clearly the pathological problem, issue with pathology, with pathologists, with the way you read the slides, with the way you define success in terms of histological efficacy. Yeah, clearly. I mean, we kind of didn't want to see the problem, but we could not avoid it once it was out there in our face. And Stephen's work on that and the paper he published together with other people from the Sirius trial were the first ones that clearly show that we have a problem. We know that histology can be variable, but here they've shown that this can have a real impact in the way you define success or failure in a trial. So, and that clearly is not just me, since the FDA now is no longer happy with just the way we did business before, and they're really asking people to think about ways to overcome that. So I would think that this would be the easiest answer to your question, but it's an interesting question.
I think back to NASH Tag last January, right? And Stephen presenting the serious results, and then there was a panel with a debate actually with you and Rohit and Stephen. And watching it and realizing that you all were grappling with this thing that was that important that just hadn't been there before. So that makes a tremendous amount of sense to me. Stephen had to jump off early, and you've been really generous with your time. We're looking forward to having you back with us again in 2021. Thanks for joining today. And Vlad, thanks for doing a tremendous job of summarizing a whole year very cogently and without a lot of time to prep for it.
Thanks for giving us the opportunity also to share with you our point of view, and also to provide these podcasts, which are very educational.
Our pleasure. More fun ahead. Or in the words of Sancho Panza, more misadventures.
Merry Christmas and happy vaccine.
This ends our extrasode with Vlad Ratziu. If you find this extrasode concept valuable, please let us know. And with that, enjoy your vacation and stay safe. See you in 2021 on the podcast.